Good morning, everybody. Can you all hear me at the back? Yes, that's good. I've got positive there. Excellent. I think we're going to try and be strict on timing, because I remember last year, the people who were here last year, it ran over. And the first people who had their networking at 11 had to rush, I think, and miss a bit. So we've given ourselves a bit more time. So hopefully we can try and do it within the 7 minutes for the presentations, which I'll try and be quite strict on. And then we should finish way before 11 o'clock, which means you can go off to your places and get yourself set up for your networking. So, very warm welcome to Pivotal Links 25. This is number 12, Mel, am I right?
12, yeah.
12th one we've done. The first one was in a little pub somewhere with about 6 or 7 people. I think some of us, hands up, who was at that first meeting? Yes, thank you very much. There we are. Here we are. So it really has developed then. We've got, it's fantastic to have you all here. Some of you have travelled as far as China, and some of us from Summertown, 10 people. So it's great to have people globally here. Thank you very much for coming and joining us. As I say, it's the 12th Pivotal Links, and we've got a very strong turnout. I think the last count was 73 companies represented and 111 delegates. So that's great. I'm really, really pleased with that. Over the next 2 days, there'll be plenty of opportunities to reconnect with familiar faces. I've already had some really nice comments saying that it's like part of family meetings now. Some people come here every year and they meet up with friends, which is lovely. So you'll meet with familiar faces, you'll meet new ones. There's quite a few new companies who've never been here before, which is great. Makes it more interesting for us to engage with you as well, so thank you for that. And I hope at the end of the 2 days, we'll hear more about the latest developments across the sector and how the market is doing, which should be quite interesting. So a quick overview of what's ahead today and the next 2 days. We'll start shortly with our popular speed presentations. And I think we've got 8 of them all sitting here in the front row. And it's a great way to discover new technologies and ideas. After that, you've got the rest of the day and all of tomorrow to network. Okay, you've all got the Brella app, I believe. And you'll have all your meetings in there. It's very flexible, so you can change it as the day goes along. But my suggestion, unlike me, where I've printed out, the very old-fashioned way I've printed out, it's already changed. So refer to your Brella app, and you'll see notifications if people want to change. There are plenty of opportunities, like a lunch break, where you can have a gap. You can have your meetings up in the lunch hall. Now, the lunch hall, if I'm right, back into the hotel reception, up the stairs, and there's a buffet lunch from, what time is that? That's from
1 till 2.
1 till 2 today, and from
12:30 to 1:30.
Yeah.
Tomorrow.
So that gives you a chance to go up there. It's very informal, and that will work very well. Tea and coffee is available throughout the day, so help yourself whenever that suits, and that's in that area there. Excuse me. For those attending the gala dinner tonight, we'll meet at the hotel bar, which I think we'll meet here. We'll meet here at 7 p.m, ready to sit down at 7:30. Dress whatever you want to wear. I mean, we're not going to be strict on what you want to wear, so whatever you like wearing. And obviously there's food and drink served until about 10:30. This year we're using the Brella Network app, as you've seen. We've scheduled all your meetings in there. On the app, you'll see there's a table plan. That's very important. So refer to your table plan. Some of you have got dedicated networking tables where you'll have your sign on it. So if you're meeting someone who's got a dedicated network table, that's probably where you'll be meeting. If you haven't got a table, you can meet, and the person you're meeting hasn't got a table, you can meet back in here, in the hotel reception, in the corridors. There's plenty of spaces that you can meet at. If you've both got a networking table, then you're going to have to communicate with each other and see which one you actually want to be on. If you're not sure where you go to the Pivotal Scientific registration booth, and we'll have someone there who will direct you. There are table plans also scattered around that you can see. I'm lucky enough to have my own little room. Okay, so I've got room. So in the hotel reception, there is a series of pods, which you'll see there. So if you're meeting with me, get to that pod area there. It's number 2, and I'll be in there. So that's where I'll be based. Before we get going, I want to say a huge thank you to our sponsors. Here you can see we're very lucky this year to have some very good sponsors here. They've all got booths as well in the sponsorship area, so please visit them because they have paid a lot of money to be here and they want to meet with you guys, so I really appreciate their support. And they play a very, very big part in making this event possible. Finally, most importantly, take a moment to familiarise yourselves with fire exits. There's some there, and throughout the building you'll see them. We're not planning any fire alarms, I don't think, so if you hear one, it's a real one. So, any member of the Pivotal team are here to support you, so reach out if you've got any questions at all. We're really looking forward to a couple of really productive, engaging and enjoyable couple of days, and it's all about networking, so please take advantage of that. Any problems, come and see me. Okay, thank you very much. Now we are going to start the presentations. And we are going to start with my colleague, Stefan Hoyle from Pivotal Scientific, and I'm going to leave it over to Stefan.
Thank you, Tim. Well read. Interesting, 7 minutes to give you my view on the market conditions and what you should do about it. If that is not a challenge, I don't know what is, apart from pressing this button and making it work.
Great.
7 minutes.
Oh, that was funny.
Where's he gone?
No, there we go.
Yeah.
Tim's asked me to do a number of things. Explain to you US economic policy and try and give you some insight on the impact on life science. That's a minute already gone. The role and the evolution of AI and machine learning. How that impacts on marketing and the capabilities you now need to consider. Commercializing science. The one biggest challenge I think we face is not the quality of science we create, but our inability to tell our customer. And finally, why Chelsea is so awesome. And decline not to comment on the last one. If you don't follow soccer, you're like me, it's a complete waste of time. Now, being a non-scientist, coming from telco and IT, it's always a challenge to think if I can tell scientists what the next evolution is in science. But actually, that's not really my point. My point isn't what's going to happen next. I'm the last person to ask. My point is, all scientists agree, change is constant. In the industry you work in, you have a clock speed. That's amazing. You are continually innovating and building new products and new services. So if we all accept that as a given, why is it that we don't change our organizations? When I consider this industry to my prior industry, we are so far behind in how we commercialize the wonderful products and services you create. So when you think about the impact of tariffs, is there something you can do about it? No, I'm not here to tell you whether I think it's right or wrong, the politics versus science, but the impact is profound. 90% of small to medium-sized biotech companies for their FDA-approved products have over 50% of imported material. The one area where we're starting to see this impact is within products and prices. And the knock-on effect to you, a small and medium-sized business, is you've got to cut costs if your revenue is declining. And where do you go first? Sales and marketing and R&D. So the impacts on tariff don't just impact your ability to generate revenue. It's the decisions you subsequently make as a consequence. We spent 2021 and 22 trying to work out how to build more resilient supply chains. One of our biggest challenges. Did we learn? Well, the problem we now face today is the disruption on supply chain as a consequence of world policy. We are one of the few industries that is truly global, judged by the number of sponsors from the different countries we operate. Yet the challenge our industry faces as a result of tariffs is our disruption on our supply chains. Even in the pharma industry, where today tariffs do not exist on finished drugs, there is the threat of 25%. I could describe this across medical devices. I could describe it across a whole variety of different components of our industry. And the key question is, who does it really hit? The small and medium-sized business. A large farmer can lift and shift its production from Europe to the USA in a flash. And what can we do if we're not USA based? We've seen some examples of small businesses create local CDMO partners so they can bypass tariffs by lifting some elements of production and the finished product being USA based. But by and large, our challenge is we have to go with the wave of tariffs that are applying. Whether you agree or disagree is irrelevant, it's what we do about it. And therein lies the debate. AI and machine learning has always been seen as being a wonderful tool to improve discovery. It improves targeting, yet it is so much more in its role that it plays within our industry. We're just not yet embracing it. And what's happening is your customer, my customer, and other businesses' customers are hesitating. Because when you don't know what's going to happen next, you pause. And what are we seeing? Declines in revenue. Now, whether you measure that against last year or your peak year, we are seeing declines in revenue. And the question is, what do we do about it? So I thought I'd give you some context around where we're seeing this impact. Trade policy is having somewhere between 5 and 10%. Now, the reason why it's lower than some of the declines you may be seeing is because service industries are experiencing exactly the same problem. Actually, what's interesting with service industries, we're also seeing growth. Because where businesses want to outsource, which is now becoming increasingly common, a capability to a third party, it's enabling service industries to drive growth. But by and large, service industries and product manufacturers are seeing declines. Therefore, distributors are seeing declines as a direct consequence. There are always exceptions, whether it's product-based or whether it's a particular application. And in the case of Neuro, that's because grant access has not really stopped. It has continued. And if you think about the customer we serve, they are often dependent upon grants. And neuroscience, they have paused less than others. In M&A, we've seen this fundamental shift from risk that sits with the buyer to risk that sits with the seller. What does that mean? I have seen a contract for sale where the seller has committed that they will hit the forward forecast numbers for every year. And if they fail to achieve it, then the penalties are far greater. So they get less for their business than they did before. And that's having an impact. We measure M&A by a factor of EBITDA, earnings before interest, tax and depreciation. And that's dropped by somewhere between 3 and 5 in the last 3 to 4 months. Actually, new ways of working is not just the fact that most of our new employees believe they can do their job from home. It's the fact that investment profiles now are milestone-based. No longer, if you go to investors, they give you all of the cash. I'd like to see some example of that translating into action before they give you the next tranche. Now, that's very specific to private equity and venture capitalists. However, the principle still applies. And then finally, AI and machine learning. How many have you seen that if you go online today and do a search for an antibody, an antigen, a protein, it is no longer the recommended or most highly supported supplier, it's an AI-generated response. In 5 months, we've gone from, let's spend all our money on Google Ads so we're top of the list, to now AI-generated response, and how many of you know how you get on that list? Because they often recommend suppliers. So it now impacts fundamentally search, and the majority of the people who buy research products do it via search. They do it via Google search. And that predicament is even more visible when you think about traditional methods of being able to data mine significantly. And 3 of our partners here today all do significant elements of data mining to derive a better answer. But I've now got examples of pharma who have replaced marketing with generative AI who build content. Content is no longer rendered by a person, it's done by ChatGPT or equivalent. This industry is fundamentally changing, and the impact of AI is profound. I found 400 companies who claim they can render a service involving AI or machine learning to give you a better outcome. Now, these vary dramatically, but I know of 4 businesses in life science who no longer use Google. They use software like rendered up here to give them the answer to the question. Someone uses Microsoft Copilot all the time. They stop using Google, they just use Copilot. But do we know where the sources of Copilot come from? And how we ensure that when they get the search, they give the answer we want them to see? Oh,
Yeah,
well, I'll move on then.
Yeah.
So what's the impact? We have got to think differently about marketing. It's much more personalised and it has to be more relational. Customers want to do more for less. You have lots of customers, but do you truly know them? And probably one of the most important components here, if you're going to survive the next problem statement that hits our industry, we've got to build a plan for it. So my summary, I don't know what's going to happen as a result of tariffs. I have no answer, and very few people do. But what I would ask you to do is think that it's not just science where you need to innovate, it's your organisation. Thank you.
Thank you very much, Stephen Hoyle. I'd like to now open this to Moa from In Vivo Biotech.
Sorry, just to have, yeah.
There we go.
So, thank you all. Tim, I'm very glad to be here. Thank you for the invitation. And when I look back to the last year, so it was my first time here. So In Vivo was almost unknown. And then I met Tim and he said, yeah, we have the Vital meeting, come here and we will meet a lot of people. And I think this is the best decision I made within my career with In Vivo. So for anyone who's here, this is the first time, believe me, that will be not the last time. So I'm going to introduce In Vivo Biotech to you. So who we are, what we are doing. In Vivo is just one of the largest Germany contract manufacturers. So we are based in Berlin or near to Berlin. I will come to that in a few seconds. We have more than 25 years experience in molecular antibody production and recombinant protein or even recombinant antibodies. For us, it is the most important thing is the high quality and giving a solution for the customers. We are ISO certified, both the 9001 and 13485, and we are GMP compliant. So we are especially for the diagnostic. So we are not GMP for therapeutic, but we are for the diagnostic. We are mostly serving the Europe market, but we started also in the North America and South America, and also Asia. Asia is like only 20% of our revenues is going there. So we founded in 1998 when we started. So it was the development of the whole factory. We started our serum, our own serum-free media. So actually what is good in Vivo, everything we do is coming from in-house. That's why we are cheap comparing for cost. So 100% German manufacturing, but comparing with the competitors, we are price, cost, good. The ISO certificate came, so we developed also the stable show cell line, and also our In Vivo vector, which we use it for the transfection for the hex cells and shows suspension cells. When we started then the cooperation with Bruker, which we started to produce for Bruker the whole consumables. That's why Bruker decided they are making a lot of money with us, decided to buy us, and since 2017 we are a Bruker company, and we started also to be IVD, so the 13485 because of Bruker products and other products. And 2024 we started our In Vivo catalog product, so we started with our name going to the market. So we are based here in Berlin, near to Berlin. We used to be only this building. Right now, this is the whole building belongs to us. We are expanding. You can see that. This building here is only for Bruker, so only the production for Bruker. Here it is the protein and antibody production, also for the immunoassay production or development. Here is our warehouse. So it is quite big. So it is a quite big campus where we can do our stuff. So if you need anything, we can produce quite kilograms from antibodies, monoclonal antibodies, and proteins. So this is not an issue for us. What our mission is really to grow up with our customers, with our partners. So this is the goal from all this business, and it is more how we grow together. For us, this is quality is the most important. And I think most of you also take care of the quality. So every company is taking care of the quality. For us, this is also one of the most important things. So anything coming from out, it go to the transit or a hotel outside the company. And first of all, to be tested that it is free from any mycoplasma. And then it goes to our production rooms. When we are looking for the cell vitality, productivity, metabolicity, and after that it goes through the production. After the production, we go for the normal SDS, SEC, and also the antitoxin level. The antitoxin level we provide always with antitoxin level 3. But here is the question, every customer has another definition for what is antitoxin free for them. So some of them, they say, I'm happy with 10 EU. Some other, they say, no, it should be less than 1. And this is, for us, it is not a big deal. So this is the service portfolio. I'm not going to go through all of them, but this is the most important, the monoclonal antibody, recombinant, stable cell line development, and epidermal cell development. We do also do cell banking. Antibody sequencing, DNA preparation, we do protein labeling, we do lyophilization, we do immunoassay development, but also immunoassay production. So if you have your recipe, just give it to us. We send it to your customer with your label, with your name. So this is also one of our services. So we can call it, we do OEM for you. Last year we started, so last year when I was here, we had 0 catalog product. Now we do have cancer biomarkers, we have also cancer antigens, we have in vivo oncology antibodies, we have isotypes, and coming soon is the neuroscience. We are in discussion with some consulting companies or also with some universities to get the best from that, but this is coming soon, hopefully this year too. So I told you about our quality. So for us, it is important that our quality, we do have some exclusive clones or antibodies which only belong to in vivo. And some of them, this is like the MSTAB or anti-mouse stabilin 1, which has been published very well. And this is the future for the oncology. We do have the clever one. This is the anti-human clever. So we have the mouse one, the anti-mouse, and we have the anti-one, the human one. And the human one has been published also very well. And the future is saying that the clever one, it will be the new BD1. And we are happy with that because right now it is in a theoretical study and it is close phase 2 with very high rate. And we are right now in phase 3 theoretical study. So some of our antibodies, because we started new with the oncology antibody, everyone is saying, you need to validate the antibody and so on. So we validate an antibody with the competitors in the market, competitor A and competitor B, and we could see that the in vivo antibody is slightly better than the other competitors, which means it's not like better that it is a big deal. So it is still, they are still in the range and all of them is working, but we have the same quality, a bit better, I would say. The same is here also. I choose only BD1 and BDL1 because everyone is talking about these antibodies. And I saw that in vivo has been always being better than the competitor. Yeah, this is all from my side. I hope that I have my 7 minutes. Yeah. Can I get 1 minute more?
Thank you, you did very, very well.
And if you need any services, we are here. So Karen and Mohammed is here, myself too. We have some meeting with you guys. But if you have any question, if you need any service, if you need any support in Europe, so we are for you here.
Thank you very much. Next up, is Ariel from Stressmark.
Thank you very much. My name is Ariel Laurier. I'm here from Stressmark Biosciences. I'm based in Victoria on the west coast of Canada, as far west as you can go in the as I was reminded yesterday, not in North America, but in the southwest of Canada. I'm here to talk to you about reaching the horizon. This is with respect to an assay that I'm going to introduce shortly in the Parkinson's space, but also in the Alzheimer's space. Now, as a brief overview, Alzheimer's and Parkinson's, you're probably familiar with these, as most of us are, but these diseases rely on the aggregation of certain proteins. And with Alzheimer's, the ones that we know about, A, beta, and tau. With Parkinson's, it's alpha-synuclein. And the bottom schematic in the left-hand corner shows you how this works. We have these monomers that are rather benign. They form oligomers. These look a little bit like footballs, although I would say for the Chelsea supporters, they look like footballs from Liverpool Football Club. These can then go on and form fibrils as well. Fibrils look like little strings, but the strings can break. And on the right-hand side here, you see a schematic of what happens in the cells. So the oligomers are generally what do the killing of the cells in the brain. In the brain. The fibrils, on the other hand, are more responsible for the disease spreading. So they go into cells, not going to worry too much about they get in and out, but they do, and they break, and then they enter other cells. Now, the key element here is that those fibrils, if we go back to the schematic here, they have this feedback mechanism, and the fibrils can catalyze the production of themselves, okay, from these benign monomers on the left. And that is exactly how the assay that we're talking about works. So we have the monomers on the left-hand side, and the seeds that are next to it in red are what we think of as the natural sample, the seed, the catalyst. They cause the aggregation of the monomers into these rather large string-like complexes. The interesting thing about that is they can then bind a very, very simple thioflavin T chemical that fluoresces. So you have a very simple assay. And what you can imagine is if you've got more seeds, you've got more natural sample, this thing takes off quicker. If you've got none, well, you don't see anything. And if you've only got a little bit, it's somewhere in between. The sample itself is interesting and relevant with respect to what I'm going to show you next, is because typically nowadays we like to use CSF, cerebral spinal fluid. Why? Because you don't want to go and stick a knife into somebody's brain, take part of it out. People don't enjoy that particular part of the process. The other element, though, is there are tons and tons of biobanks around the world that have samples that could be decades old that are formalin fixed and paraffin embedded. Now, typically you'd say, well, so what? Well, it's a treasure trove of information on what has gone before. And these could be natural samples that have been recorded, taken over decades. And those are samples that we can also use in this assay. So on the left-hand side, you see an assay. This is, of course, with a competitor product, not ours. But it's not atypical of what you would expect in a failed assay. The reason is that this preparation that you see on the left-hand side from our peptide is not actually designed for this assay at all. The problem with this assay, and why it's been so difficult, is because the actual substrates themselves want to aggregate. That's what they do for a living. Okay, so you have to have a very, very specific assay that is able to respond to the seeds and aggregate, while at the same time trying to stop the self-aggregation of the process. So you've got a bit of a push me, pull you thing going here. Now, if I go to the right hand, oops, went too quick, wrong button here. If we go to the other side here, this is a preparation that works very, very well. This is some work that we did with the University of Oregon, and you can see some really nice discrimination between the blue, which is the Parkinson sample, and the red, which is a control. Even more interesting is the fact that it's a gut sample, not a brain sample, not CSF, but it is also from a paraffin, a formalin-fixed paraffin-embedded sample from a bank. And you can see this incredible discrimination that you can get, so you can show that the disease is present. Going to CSF, in blue you see just a recombinant addition of seed material, so that's a very quick takeoff. But in red, you have the Parkinson's CSF, a Parkinson's patient, and in black, the healthy volunteer. And you can see, again, this incredible shift. It's a very, very, very clear, easy to read signal. Moving on into Alzheimer's, which is an even bigger, roughly 7-8 times bigger market. For the first time, and this is data that nobody in the world has ever seen before, we actually see, again, from formalin fixed paraffin embedded tissue in the gut, we can discriminate the blue, which is Parkinson's patient, and the controls, which are the other two. And this isn't optimized really fully at this stage. Going to the brain, it's even easier. These are 3 different concentrations, so it doesn't even matter. But you again see in all cases you get really, really good discrimination. So at this point, I just want to announce one thing that has been a big deal for us. We have just signed an agreement with the Michael J. Fox Foundation for Parkinson's Research to provide worldwide the distribution of the alpha-synuclein substrate for the Parkinson's disease. The Alzheimer's is a different thing that we're doing ourselves, but we plan to kick that off sometime towards the end of this year in Q4 of 2025. Thank you very much.
Very well. Thank you, Ariel, for that. Now, next we have Brian from Apoligo, a brand new startup company.
Just move ahead. First of all, I would like to thank Tim and the Pivotal team for having us here to discuss Apoligo. It truly is an honour and a pleasure. The other thing that I just wanted to highlight is our chairman's here as well, Nick Hutchins. So if you want to have a discussion with either Nick or myself, please reach out. As you know, scientists these days are under immense pressure to publish lots of scientific publications, as well as accelerate their drug discovery processes. And therefore, they're looking for assays which give them more data points per assay, rather than, as well as using less samples. And therefore, this is whereby they turn into multiplexing. So instead of doing this, where's the pointer?
Pointer's
In the middle.
Oh, perfect. Instead of using the standard assays, which is immunohistochemistry or ELISA, they're all, as I said, transitioning towards multiplexing. So one example of that is actually the proximity extension assay, whereby a scientist can study from a single drop of blood 5,000 proteins. So that we're moving into a new era, as Stefan highlighted earlier on, where multiplex will be the mainstay moving forward. Now, we believe the major tool that helps drive these multiplexing assays is actually antibody oligo conjugates. And this is whereby Apoligo believes we've got a unique offering to the market. And let me just explain what I mean about why we believe antibody oligo conjugates are the ultimate research tool. If you look on this Venn diagram, on the left-hand side, what you see is lots of oligo fragments, which allows scientists to study the genome or the transcriptome. On the right-hand side is the space that we all operate in, whereby customers are able to analyze proteins due to the antibody specificity and binding affinity. Now, if we combine the two, we're in a different, powerful world. This is whereby you benefit from the protein specificity of the antibody, cross-linked with the unlimited readout potential of the oligo. Therefore, it's only a matter of time for someone is able to analyze the whole of the proteome using antibody oligo conjugates. As I said, at the moment, all link are up to about 5,000 conjugates per sample, and they're adding more antibody oligo contents moving forward. The other thing to just highlight is that the multiplex space is obviously very exciting at the moment. And what you can see is we had the recent acquisition of Olink by Thermo Fisher, which was for $3 billion. And what's nice about Olink's technology, it's all driven by antibody oligo conjugates and proximity extension assays. So this is a field that we all need to be moving into. I think it's a real differentiator for us as an industry. And there are other players emerging, are already operating in the field, NanoString, 10x, Alamore Biosciences, the list goes on. The other thing is I would really encourage you all to go and have a look at the Nomics website. They've got a really elegant assay which highlights the benefits of their technology in multiplexing. And I think that's going to be, they're going to be quite an interesting company moving forward. Now, as an industry, we've done very well in generating lots of antibodies. The last time I looked, there was about 7 million antibodies on the CiteAb database. But when I actually look into the market of antibody oligoconjugates, there's 10,000. So I think there is an opportunity for us to be able to really support the customers when they start transitioning over to multiplex assays. Now, as an industry, we offer various different solutions, whether that be antibody oligoconjugate products, alternative options such as custom work, which unfortunately they get out and access 200 times excess material. The other is DIY conjugation kits. But the challenge with that is that unfortunately that they are time consuming. And one thing I just wanted to highlight about the profile of a customer who's doing multiplexing is this. In the future, they're going to need lots of antibody oligoconjugates, but they're not going to need per conjugate itself, they're going to need less material. So 10 micrograms is probably enough for them to either publish a paper or to do their PhD. So that's benefited due to the amplification of the oligo strand itself. So again, we've got to start thinking differently as an industry, trying to focus on selling 100 microgram vials and get repeat businesses in the future, I think is not going to be the long-term solution. Now, when it comes to the antibody oligoconjugation space, the main players out there, whether it be NanoString and Car, have done amazing jobs in generating antibody oligo content, but they're only operating somewhere between 1,000 conjugates or 10,000 conjugates. If we were going to study the whole of the proteome, we've got to get up to 20,000 different conjugates. Factor in that you also got various different post-translational modifications, the numbers increase exponentially. So there is going to be demand moving forward. And this is where our oligo solution is. What we intend to do is to hold a library of oligos as well as a library of antibodies, which we'll hold in stock. And we will only make a batch when the customer places an order with us. So if they select the pair that they're looking for, we will provide that product to them. The other thing is we're going to be changing the scale that we manufacture. A batch run will be 10 micrograms. So it's not 10 milligrams, it's not 1 milligram, it's 10 micrograms. We're right down at the level that customers are looking for to be able to for the right level of antibody the right level of antibody oligoconjugate that we're looking for. The other thing is we're going to do a QC batch release based upon our proprietary method, which guarantees that we're able to deliver batch consistency to these customers. So whether they buy a batch today or in 10 years' time, as long as it's the same pairing, we'll be able to produce the product consistently. The other thing is we're going to be able to dispatch this to the customer in less than 3 days. In regards to the whole process and our proof of concept, we're actually going to automate it. So the idea is whether we have to do 10,000 conjugates today, 100,000 in a year's time, and 1 million in 3 years' time, we'll be able to do it. All we've got to do is buy extra capital equipment. What I'm proud to see is the technologists that are driving our platform are Nick G and Nick Knowles, who have over 40 years of conjugation expertise. On top of that, they were the inventors of the Lightning Link technology, as well as, and obviously they were both contributed to the sale of Innova Biosciences. So we have a high level of confidence that we'll be able to deliver this platform. The final piece is that once we're ready to deploy the platform, that we'll have 4 different business models to be able to do it. I'm not going to go through all 4, but the point that to people in this room, what I'm really interested in is getting access to antibody content so that we can get that new antibody content onto our platform. So what we're going to have is an unstocked model whereby we enter into a commercial license to be able to access your material. Customer places the order, you ship the products to us, then we get the product out the door. The other is a consignment stock model whereby we hold stock at our facility, the customer places an order, we make it very quickly, and then it's dispatched within our 3-day time frame. Hopefully, the consignment model will be of interest to people. Because the other thing that is, as I said earlier on, we only need small amounts to be able to generate and do a batch run. So in summary, Aboligo is a really exciting adventure, and I hope that we can partner with the various different antibody suppliers in the room to get your content onto our platform. Furthermore, I think it allows everyone to move into the multiplex space, which is really hot at the moment and can only continue in that direction.
Thank you, Brian.
Thanks very much,
next up is Steve from Telerix.
I pronounced that correctly.
You did this time, thank you.
So, biosample logistics, probably the least interesting of topics you're going to hear today on the face of it. But sample logistics, life samples logistics are fraught with a number of complexities and causes a lot of anxiety. You can either freeze live biosamples, so cryopreservation, but this has a lot of biological incompatibilities and logistical complexities. Or you can use biosamples fresh. But the problem with using biosamples fresh is that they're subject to a very short shelf life. So it causes a lot of anxieties that a lot of people are familiar with. Results can be skewed by artifacts due to shipping, due to poor sample quality. Samples can get stuck in customs. That happens a lot. I'm sure everyone has experienced that. And what you end up with is high-stakes shipments. You're gambling with your results and project success. You get project delays because of more samples. You have samples that arrive thawed, and really it causes a lot of issues. And additionally, cryologistics is very, very carbon heavy and it's killing the planet. So there is another way. We have a solution which is a cryo-free, hassle-free ambient logistics solution. It extends fresh sample shelf life from hours to weeks. And what this does is this enables complex tissue models, sensitive cells, and therapies to be shipped. We have products which are supplied as kits. They're incredibly easy to use. All you need is a pipette. In some cases, you don't even need a pipette. And what these products do, they ensure that sample viability and functional performance is maintained. They're easy to validate. They de-escalate shipping. They make it a lot easier compared to cryologistics. And they can slash costs by up to 10-fold compared to cryo. So what this does, it offers a flexible, low-cost supply chain and workflow from sample collection to final application. So how does it work? So it's all based around hypothermic preservation. If you think of cells or tissues in your body or in an incubator, they're at usually 37 degrees. Normothermia is in the range of 32 and 37 degrees. And at these temperatures, cells are metabolically active. When they're metabolically active, at these temperatures, they have a fluid membrane. The idea behind hypothermic preservation is you reduce temperature, you reduce enzymatic activity, and therefore you reduce metabolic activity. Cells become paused in this state because they don't have the energy to do anything else. And in principle, this allows you to store cells or tissues within a sealed vial without the need for metabolic substrates or gaseous exchange. In essence, you can just ship them in a tube. But the main problem with doing this is that that fluid membrane turns into a static membrane. This results in pore formation. You have uncontrolled entry of ions of water into the cell. They burst, they die. What our technology does, we have alternate-based hydrogels, specific formulations of alternate hydrogels that we've developed over time, which actually stabilize that lipid membrane. So what that does is it prevents unregulated transport, prevents the continued deterioration of lipid membranes, and prevents cells dying. And because it works on lipid membranes, it's relatively universal. It works on tissues, cell models, viruses, LNP vaccines, blood products, essentially anything that has a lipid membrane component. And this means that you can extend the shelf life for up to 2 weeks without the need to freeze the samples. And it's very, very simple. We have protocols where essentially you just add your sample to our gel. It gels in situ, then you can seal the tube or bag or whatever format it's in to the destination, and then when the user needs it, they just add a buffer to dissolve the gel back to a liquid. So very, very straightforward. And we've optimized this technology to be suitable for a number of different product formats. depending on the biological specimen you are preserving. So we've got cytosol for cells and viruses. We've got tissue ready for tissues and organoids, blood ready for whole blood, leukostore for leukapheresis, and well ready for in-plate models. And well ready is really kind of suited for shipping ready to use assays. So this might be ready to use organoids, ready to use 3D models, etc. So just to conclude, we have a solution which can be 10 times cheaper than cryopreservation. So this allows you to keep money for science, not for shipping. We've modeled that it can be up to 99.7% fewer carbon emissions compared to cryo logistics. So you really have an impact on the planet by shipping ambient. We have zero compromise. We have a huge amount of validation across over 80 biological samples now, showing that we can maintain viability, function, and biological integrity. You move any sample anywhere. You don't need freezers, you don't need dry ice, you don't need dry ice top-ups at customs. Really, it makes it a lot less stressful. And it's ready to run. Simple, fast, and seamlessly integrating into SOPs. And that's all I'm going to talk about. Helen talked to me. You asked me why there are a load of hedgehogs all over the slides. There is a reason, and it'll be good to chat. We're also 8 years old today, so if anyone wants to buy me a drink.
Thank you, Steve. I love the branding on that, by the way. Next one up is Jonathan from Cavidia. Here we are. Have I pronounced that right, Jonathan?
Sorry?
Have I pronounced it right?
Yes, Kavidi.
Cavidia, there we go.
Thank you very much. Okay, nice to be here. My name is Jonathan Royce. I'm the Director of Business and Applications Development at Kavidi. We are a small, I usually say 17-year-old startup. We went through a major transformation in 2020, which has led us to where we are today. And we have developed a technology, it's an add-on technology for any immunoassay called Exozyme. And I'm going to walk you a little bit through how this technology works, show you some examples, and I'll wrap it up. So Exozyme is an add-on technology for immunoassays. It's based on a technique that we call BOLD, binding oligo ladder detection. This is a technology that we have patented in Europe. It's patent pending in the US, although we expect that patent to be approved this year. It provides up to 100x signal amplification and 200x improvement in sensitivity. It's very cost effective. It doesn't require any new equipment. It allows researchers to use their validated antibody pairs. And really the only price you pay for this is a minimal extension of the assay time. So I'll show you how this gets integrated into a standard sandwich ELISA. But this can really be integrated into any immunoassay that's based on some sort of ligand analyte detection. So in a standard ELISA, of course, you have your capture antibody, which is coated onto a surface. This can be a plate. It can also be bead-based technologies. You then add your sample where the antigen is bound. This is, of course, familiar to all of you. And then you add a detector antibody. And in our case, we do not add a biotinylated detector antibody. We add a detector antibody that we have conjugated with a oligo-DT primer. End users do this conjugation themselves today using our Exozyme conjugation kit, or for 3 analytes where we have a lot of demand for cytokines, we have now launched pre-conjugated detector antibodies, or pre-conjugated pairs where the detector antibody has been conjugated based on actually antibodies that we source from the team at biotech. After this, the next step is to use our polymerase reaction kit, where we add a room temperature enzyme, we add a template, and we add some BR brominated UTP. And what the enzyme then does is it builds out a oligo strand, a DNA RNA hybrid strand. We then do a wash step to remove any of the remaining reagents. And then we add a tertiary antibody, which is highly specific for this oligo strand. Today we offer this antibody, this tertiary antibody in a biotinylated form, which allows people to use whatever sort of reporter molecule that they want. We also provide pre-conjugated APC and PE antibodies for people that are using either, for example, SMCX Pro or Luminex platforms. And then you generate your signal, and of course what you can see here is that you get a much higher molar ratio of your reporter molecule to your analyte than what you would get in a normal sandwich assay. Here are some examples of ELISAs that we've developed in-house, where we first developed the standard ELISA, and then we have applied the BOLD technology to amplify the signal. So this is sort of different categories of analytes. In the upper left, you have infectious diseases. This is where we actually started in 2020, which was trying to detect spike protein for COVID. And here we were able to improve or reduce the limit of detection by a factor of close to 50. In the lower left, you have a typical cytokine inflammation biomarker, IL-4. This is one of the ones where we've launched a conjugated antibody pair. And here we also achieved about a 50x improvement in LOD, getting down to sensitivities in the femtogram per milliliter range. We always get the question if this works in serum. So in the examples on the left, you have samples that are in PBS and casein, but on the right, you have, in the upper right, we have one of our examples where we've worked with serum. This is a cardiac biomarker called troponin. And this is actually the best example we have to date where we improved the sensitivity by a factor of close to 200x. And we were able to get down to the 50 femtogram per milliliter level on this assay. It's a little bit of a unique assay where we have 2 capture antibodies coated on the plate, and then we have a single detector antibody that carries our oligo DT primer. And then in the lower right, you see an example, which is one of the areas where we're most active, which is in neurological research. Here we worked with p-tau181. We've also worked with p-tau217, NFL, and amyloid beta, but this is the example I chose for this presentation where we got about a 60x improvement in LOD down to about 80 femtograms per milliliter. I always get the question as well, is it repeatable? Can we validate this technology? The answer is yes. We've done some studies on another cytokine. This is on TNF-alpha, where we've looked at both intra and inter-assay precision. You can see in this table that the samples are organized from high to low concentration, and you can see that in the ELISA case, in the standard ELISA, we start to lose basically repeatability in both cases because we're down at the limit of detection, but the BOLD assay stays quite nice down below 10% in terms of CV. The technology is quite easy for users to access, so we provide the technology in kits for research-only use. There's an exozyme conjugation kit, which allows them to conjugate their detection antibody. There is then a polymerase reaction kit, which allows them to perform the polymerization step. And then there are 3 different detection kits, depending on what they're using as their reporter molecule. I'll just mention before I close that the examples I showed today are on ELISA. We've also applied this technology to techniques like gyros, SMCX Pro, MSD, Luminex. It is actually very versatile in terms of its applicability. We, as a small company, work primarily with ELISAs in-house because we don't have the resources to acquire instruments that can enable these other technologies I mentioned, but we have a lot of collaborations with customers and research universities now that are using those automated systems and applying this. So I'm the only representative from Kaviti here this week, so feel free to reach out to me if you want to know more, or you can contact me with the info on this slide.
Thank you, Jonathan. Thanks to Jonathan. And the last but not least, is Aham from Epigenics, which is another new startup company.
Yes, there we are.
Hi, everyone. Hi everyone. Thank you, Tim, for the introduction. It's a pleasure to be here. Today, I'm presenting a novel multiplexing and antigen display platform that really has the potential to transform the diagnostic sector. This is only a small part of it, so if you want to find out more, you can talk to me, or our founder, Thibaut Wang, is there. So let's start. At the heart of the innovation is a plasmid construct that has 2 bioengineered domains, D1 and D3. Any sequence of interest can be inserted between the two domains. It can range from peptides to full antigens. Following transformation and protein production, you have a chimeric protein, inserted peptides. There are separated by, or antigens are separated by flexible linkers so they can support proper folding. And the magic happens is when you transfer this complex protein into a folding buffer, the D1 and D3 arms self-assemble into a stable scaffold that optimally display the inserted peptides. The scaffold itself facilitates expression, seamless multiplexing, and native-like display of peptides. So, what's so unique about our scaffold? We have bioengineered a surface binding domain that binds to testing surfaces like ELISA plates or nanobodies, nanoparticles with high affinity. This ensures correct antigen orientation, which is a major factor that significantly impacts diagnostic performance. We also done something very nice. We bioengineered an in vivo biotinylation tag. What this does, so you have in vivo biodylation done inside the expression system, but you can produce biotin labeled antigens without the need for extra processing steps. The result is reduced cost, improved performance, reduced batch-to-batch variation. And there are more properties. The scaffold facilitates the expression and display of short peptides, long peptides, subunits, and full antigens. It also seamlessly expresses antibody fragments and nanobodies. It can support the expression of up to 1,000 amino acids, and up to 15 peptides can be multiplexed on one construct. It is, of course, compatible with the leading technologies like Luminix and microfluidics, but importantly, it's compatible with the gold standard platforms like ELISA and lateral flow, making the technology R&D friendly, it's clinically scalable and globally deployable. And all of this is achieved with over 99% efficiency, high yields exceeding 200 mg per liter, and exceptional stability. Now, as far as we know, there is no platform that can blend all these capabilities in one system. Now let's take a real-world example where we use the scaffold. Lyme disease is a complex tick-borne infection that is very difficult to diagnose. The clinical reality of this disease is very challenging. Low sensitivity in the early stages, cross-reactivity with other infections and autoimmune markers, and a two-tier testing strategy that is slow, expensive, complex, and labor-intensive. Why is it so difficult to diagnose Lyme? This is because actually there are over 8 distinctly different Lyme-causing species. There is no multiplexing platform that can combine these strains and inconsistent antigen orientation. To date, there is no platform that can address all of these challenges at the same time until, of course, using our scaffold now, which is what we did. We have developed a comprehensive Lyme assay which combines 24 full antigens plus 120 multiplex epitopes from the 8 clinically relevant Lyme species. Only our platform's unique properties can make this possible. When we validated the assay in the UK Lyme reference lab, the results showed 16.6% improved sensitivity in acute Lyme, 23.1% improved sensitivity in early Lyme, a specificity of 99.5%, nearly 10% higher than any commercial assay. This Lyme test is a game changer because it dramatically improves early detection, it improves specificity, and it has the potential to replace the two-tier testing with one-stop test. This work is published if you are interested in further details. The great news is that this platform is not limited to Lyme. We are expanding the scaffold to work on autoimmune diagnostics. We are currently developing a type 1 screening test for diabetes. We are also developing a point-of-care test for primary diagnosis of diabetes, which can be used in GP clinics, can be used in pharmacies, even at home. We are also extending the scaffold into systemic lupus erythematosus and Hashimoto disorder. And perhaps most excitingly, currently this same scaffold is being trialed for oral vaccine delivery and to be used as an adjuvant. With that, I would like to thank you for your time and attention. Please, if you have any questions or would like to collaborate, you can contact me or Tiwi. Thank you to Pivotal for helping us grow the business from 30,000 to over 10 million pounds now. And of course, our collaborators and great team. Thank you.
That was really, really good presentation. Actually, all of them were really good presentations, and you'll be glad to see it's 10:33, so we've got time for coffee and tea before your first presentations. Very, for your networking meetings, very important now. You've had those 8 presentations. You haven't had a chance to ask questions because we won't have time for that, but they're available all day long. You've either got meetings with them or just grab them now for questions, because I think that they did stimulate lots of conversations. So thanks for your attention, everybody, and off you go and enjoy your networking.